better get going, hey?
It's about time. It's about time. It's not.
Yeah. Morning, everyone. And again, good morning, everyone. And somebody somewhere should see that I'm trying to talk. Okay.
Yeah, Tom?
Okay, good morning, everyone. Morning, everyone. I'm delighted to start the first session of today. The first speaker will be Christian Machens. So Christian has studied physics, first in Germany and then in New York, and then did a PhD in neuroscience in Berlin, and moving on to the postdoc in Cold Spring Harbor. And since 2007, he's been in the École Normale Supérieure, and about 10 years ago, he moved to the beautiful Champalimaud Center for the Unknown. And over the years, Christian has done a beautiful combination of both data analysis and modeling, where the data analysis has been extremely creative in developing new tools as needed to answer the questions that arose both from the modeling side and from the data analysis side, things such as DMX PCA and understanding how and why neurons spike. So I'm very happy to have Christian here and looking forward to the talk.
Okay. Can you hear me? So thanks, Omri, for the introduction. and thanks to Laura and Tim for giving me the opportunity to talk here. I'm going to talk about the geometric way of understanding spiking networks. Before I start, I want to point out that most of the work I'm going to talk about is not due to me, but it's due to various people in the lab. And I want to just highlight my long-term collaborators, Sophie Deneve, as well as for the purpose of this talk, Nuno Cosyne, Sander Keming, William Podlaski, and Alan Mancoux, who all have been very instrumental in helping develop this geometric framework. And I want to start by pointing out a dichotomy in the field. So usually when we try to model something in a neural network, a computation, such as decision-making or memory or object recognition, we will usually resort to rate networks. Now, rate networks are networks in which neurons are analog neurons. That means they receive analog inputs, such as the firing rates of presynaptic neurons. They weight and sum them, and then they have an analog output, a firing rate. There's usually only one type of neuron, and we can train these networks, but we use non-local learning rules, such as backpropagation, etc. But they're the model to understand computation in the brain. And they've been incredibly successful in recent years, as we know through the deep learning boom. Of course, they're not biophysically realistic, and we know that there are many differences in real circuits. So real circuits, of course, have spiking neurons, and spiking neurons fire at very low rate. They usually fire at rates of 2, 3, for instance, hertz in rodent cortex, for instance. There are different types of neurons. There are excitatory neurons, inhibitory neurons, and those fall into different subgroups. Learning rules are usually local. You know, plasticity is just local. It's only pre- and post-synaptic information that you have at a synapse, et cetera, et cetera. So it could be a long laundry list of things that are different in realistic networks. When we model such networks, we will resort to biophysically heuristic spiking networks. And now we have a problem, because traditionally, the first thing that goes out is our ability to compute anything. So we have this dichotomy between these networks that we can use to compute something that work quite well, and the networks that are supposed to be biologically realistic, but often they don't compute anything. So why is that the case? So I think there are two perspectives we can have on this. So one perspective is to say that really evolution can't invent something like rate networks because they are biophysical, they're physical constraints. You can't have long-range communication of analog signals without being flooded by noise. So in this way of thinking, it's really just about implementation. The brain has to resort to tricks to implement these types of rate networks. You have to pool neurons that are noisy to be able to build a single rate unit. It may be difficult to have neurons that have both excitatory and inhibitory outputs, so you group them into different types, et cetera, et cetera. So it's really all just implementation in detail. We've understood computation, and this is really just about implementation. Another way of thinking about it is to say that, well, no, actually, the brain does something different from a rate network, but we've managed to approximate that type of computation by rate networks. Now then the question is, what is it that neural circuits are actually doing? So I want to ask you to humor me for the rest of this talk and sort of think that maybe there is something that we actually don't really understand. And what I'm trying to build is like a different perspective of how to compute with spiking networks. First, I want to point out, though, that to bridge this gap, there has been a lot of work over the last 10 or 20 years to bridge the gap between rate and spiking networks. And there's been very exciting research in the last years, especially about the question of training spiking neural networks, from instance, the labs of Wolfgang Maas or Friedemann Zenke and others. And so now we can build computations into spiking networks. But my talk is going to be about something else. My talk is going to be about understanding how spiking networks compute. Can we understand principles of computations in spiking networks? And so I want to first start with something that we're actually very good at, and that is to read out from spike trains. So that is something we have understood very well. We can take, for instance, a bunch of simultaneously recorded neurons, take their spike trains, and we can apply something like a linear readout, filtering of the spike trains, linear summation, to retrieve interesting signals in the brain, such, for instance, the movement of an arm that the monkey may perform in a task, such as a motion of a visual stimulus or the concentration of an odor, et cetera, et cetera. And we do that usually using linear methods. So we use linear decoders. We may use principal component analysis or factor analysis or many other different methods. They're all just kind of linear readouts of this spiking activity. So that we understand quite well. But what about the inverse operation? What about the question of going from all of these analog signals to spike trains? At some level, that is a problem that brain circuits obviously face. But how do we solve that problem? Well, usually when we solve that problem, we resort to a single neuron solution. So we think of it as a single neuron problem. We say they're analog signals, and we pass them through maybe an integrated fire neuron or Poisson process, etc., and then we solve the problem. But that type of thinking is exactly what leads us to the rate networks. So maybe the problem is that this move from analog signals to spike trains should also be considered a population-level problem. Not a single neuron problem, but a population-level problem. So what do I mean by that? Well, if it's a population-level problem, it means it's a circuit problem. It's not a problem of a single neuron. It's a circuit problem. And I want to study that circuit problem in a very simple example, which is an autoencoder. An autoencoder is a system where you put in a bunch of signals. In this case, these will be analog signals. Then we're going to produce a bunch of spike trains such that afterwards we can read out the signals that we put in. Okay? So that'll be the task I want this network to perform. Now, we've studied this system for quite some time. And we've shown how to calculate the optimal connectivity in this system. We've shown how to learn the optimal connectivity in this system. What I want to show you today is that there's a geometric view of what this system is doing if you build it. So to do that, let me start actually with a single neuron. And let's just look at the problem of encoding two analog signals into a single spike train. So here, this is our neuron. It receives two inputs, x1 and x2. And then it's supposed to produce spike trains, one spike train. And then those spikes are being fed into two readout units that are going to read them out, y1 and y2. And they read it out with decoding weights, d1 and d2. Now, what happens if this unit tries to fire a spike? Now, if that unit fires a spike, so it adds a new spike to the spike train, then that new spike will lead to a change in the readout. And that change is shown here. Now, that change in the readout can also be visualized just in the space of readouts or input signals. And that's what I'm showing here. So here you have the readout y1, the readout y2. This black dot, that's where the readout is at this particular point in time that we're just looking at. And x is where this signal is. Now, ideally in the autoencoder, the readout should match the signal. So it should try to get as close as possible to the signal. Now, when this neuron fires, though, it's going to move the readout in a direction that is given by its two decoding weights, d1 and d2. So what you can see is that if the readout is up here, it's actually not such a good idea for this neuron to fire, because it's going to move the readout further away from the input signal. On the other hand, if the readout is down here, well, now it's a good idea for the neuron to fire, because this way, overall, the readout will be closer to the input signal than before. And by these type of geometric arguments, you can sort of see that our readout space divides into two regions. One region where the neuron should not spike, which is shown up here, and one region where the neuron should spike. And the line that distinguishes those two regions, that is essentially going to be the threshold of our neuron. That's going to be the point at which it's supposed to fire. Now, when the neuron fires at this threshold, then it's going to move in the readout into that direction. And the readout is then going to move to another line, which I'm going to call the reset. And these words, reset and thresholds, they're supposed to evoke the voltage of a neuron. Because indeed, what we're going to do is we're just going to define the neuron's voltage as moving in this direction here. So from the reset to the threshold, etc., above threshold. Now, mathematically, to have the voltage do that, all we need to do is we need to make the voltage compute the dot product between the decoding vector d and the error, which is the difference between the input signal x and the readout y. If it computes this dot product, then it will be exactly what I'm showing you here. Now, biophysically, what that means is that what we need to do is we need to take the readouts and feed them back into the inputs and subtract them, so that what the neuron effectively sees is not the input signal, but rather the difference of the input signal and the readout. Now, when the neuron fires, the readout is going to move to the reset. And afterwards, we're going to assume that the readout slowly leaks towards 0. Because if the neuron is not firing, we just assume there's going to be an exponential decay towards 0. Now, one neuron can only contain the readout so much. So we need more neurons to make sure that the readout does not escape the signal. So what we're going to do is we're going to introduce a second neuron. And this second neuron is going to get a different set of decoding weights. That gives us a decoding vector D2. But it's doing the same type of computation. It's going to project the error, x minus y, onto its own decoding vector D2. And it thereby creates a different threshold in this two-dimensional space, which is shown here. And if we take even more neurons, in this case 5 neurons, then we can now make sure that the readout can no longer escape the signal. So it's going to be bounded around the signal in this pentagon-shaped object. I'm going to introduce even more neurons, and we're going to call this object, this almost circular object, the error bounding box, because it's bounding the error. And to illustrate how that works, I'm just going to simulate it. So the black dot that you're seeing here is now the readout. The readout is always trying to leak towards 0, but doesn't manage to leak towards 0 because this neuron here is going to fire and is going to kick the readout right back into the bounding box. To show you what happens over time, I'm now going to also plot the input signals and the readout on this plot against time. So the signal is here gray and the readout is black. And I'm going to plot the spikes that are fired by this network. There are 12 neurons in this network, and the spikes are just colored the way the thresholds are colored. And so now we have a system that is going to make sure that the output, the readout, is as close as possible given to the input signals. What I'm going to do is I'm going to zoom out, and I'm going to show you what happens if we now start changing the input signal. So far the input signal was constant. Now we're going to have the input signal be a sine and a cosine. So this signal is going to move around in a circle in the readout space. And what you can see is that the readout will always try to leak back to 0. But since the circle is moving, 0 is now at different locations, and different neurons will take care of bounding the readout and making sure that it stays close to the signal. And so this way, we've built the autoencoder. We've built a system that is able to track an input signal, and we can visualize it in this space of readouts. Now, what if we didn't have 2 input signals? What if we had 3 input signals? If we had 3 input signals, then each neuron's threshold would become a plane in a 3-dimensional space. And the intersection of all of these planes will give us this weirdly shaped object, basically an error bounding box in 3 dimensions. Looks a bit like a soccer ball. And when we simulate it, we see basically now this black dot is the readout, and it's going to hit different neurons, different faces of this bounding box. Every time one of these faces or thresholds is hit, that neuron is going to fire. It's going to make sure that the readout moves back into the bounding box. In this way, we now have a system that is also able to track 3 dimensions. And I'll leave it to your imagination to think what is going to happen in higher dimensions. So let's summarize what we've done so far. So we basically made 3 assumptions. The first assumption is that we're going to build this autoencoder, that we're trying to map analog inputs, a set of analog inputs, onto a whole bunch of spike trains so that we can afterwards reconstruct those inputs. We've assumed that the way the reconstruction works is via a linear readout. So filtering of the spike trains, in fact, with an exponential filter and then summation, which is the type of operation that we usually perform whenever we analyze some data. We also assume that spikes are only fired when it's absolutely necessary. So they're all fired for a reason, the reason in this case just being to contain the readout around the signal. From these assumptions, we've obtained the following results. So we've seen that what we need is essentially negative feedback of the readouts to perform that. We've seen that the voltages effectively measure projections of the coding errors. And we've seen that we can visualize the dynamics of the system in the readout space. Now, of course, a critique can come in and say, well, those were nice movies, but seriously, what does this toy model have to do with biology? And so I want to, first of all, just emphasize the importance of toy models in neuroscience, because they allow us to conceptualize and to visualize things that are otherwise very complicated. And I want to show that we can visualize certain properties of neural systems with this very simple toy model. So to do that, I'm going to first point out that there's a relation between the excitability of a neuron and its position. It's the position of its threshold in this readout space. So if we make a neuron slightly less excitable, for instance, then what happens is that the threshold will actually move out of the bounding box. If we make a neuron more excitable, the threshold will move forward. It will move into the bounding box. So the first thing I want to show you is that old problem of trial-to-trial variability. So trial-to-trial variability refers to the fact that if you, for instance, record from a sensor neuron and you present the same stimulus over and over again, that neuron will fire almost like a Poisson process, okay? It will fire different spikes on each individual trial. So how can we understand that? So to do that, we're going to look at this bounding box example now with a couple more neurons. And we're going to look at only one neuron, which is this red neuron that is highlighted here. And what I'm going to do is I'm going to play the same stimulus 10 times, and I'm going to record the spikes that this neuron fires over 10 trials. So you see the readout moving inside the bounding box. Every time it hits the red neuron, that red neuron is going to fire. Now, the first thing you can observe is that that red neuron will not fire on exactly the same time points in each of these trials. And now the reason for that is twofold here. So one reason is that when the neuron fires will simply depend on the details of the trajectory of that readout inside that bounding box. The other reason is that I introduced just a very little bit of noise here, noise that is too small to cause any real trial-to-trial variability in single neurons. But that noise has led to some kind of jiggling of the thresholds here, which some of you may have seen. And so it will also depend on whether this red threshold is just slightly moved into the bounding box or slightly moved out of the bounding box. And so what is really happening here is that when this network is encoding this input signal into spike trains, there are many possible population spike patterns that will all give essentially equivalent readouts in the sense that the readouts are contained around the input signal. If we only subsample the system by only looking at one of the neurons, then we don't see this full spike pattern, but we only see one neuron, and that neuron will then look very noisy. But at the same time, the whole system works very reliably. Another type of insight I want to show you has to do with robustness. So work in recent years has shown that neural systems can be incredibly robust to the silencing of neurons. So there's some nice work, for instance, from the labs of Shao and Karol Swoboda, where they've showed that you can silence a part of the anterior lateral motor cortex in rodents for 200 milliseconds during a delay period, and then afterwards, activity will just come back. It will come back to where it was before. There's some nice work from the lab of David Dupré, where they silence the place cells in the hippocampus. So essentially what they do, in a specific environment, they inactivate all of the cells that are active in that environment. So they eliminate the place cell code. But what they discover is not that the place cell code is gone. What they discover is rather that cells that were previously silent suddenly become active and become place cells. At the same time, other groups tell us that nervous systems are extremely sensitive to excitatory inputs. So a lot of work has shown that you can stimulate just a few neurons in cortical systems, for instance, and that will lead to a perceivable change in motor output, or it will lead to a perception of that animal. So there seems to be something of a paradox here. On the one hand, neural systems are supposed to be very robust to inhibition. On the other hand, they're supposed to be very sensitive to excitation. So of course, these are different systems, they're different tasks, different animals, different labs. So maybe it's just, a matter of biological complexity. Another possibility is that it tells us something fundamental about how circuits are working, how neural circuits are working. And I want to show you that we can actually easily understand these effects inside the bounding box. So first, what I'm going to do is I'm just going to inhibit half of the population in this bounding box. So to inhibit half of the population, this population is now shown here with these thicker lines. So what's going to happen if I'm inhibiting them is that thresholds are going to move back. So see now all the thresholds move back. But you also see that the system keeps tracking the input signal. And that is because there are still neurons left that will make sure that the readout does not escape this bounded area. So there's just a very small, almost unperceivable change in the tracking. But essentially, the system just works. So it's able to instantaneously compensate the loss of some of its neurons. Similarly, let's imagine that we have a system where some neurons are slightly more excitable than others. Now, the neurons that are slightly less excitable, their thresholds will be just moved a little bit out of the bounding box, and they're shown in black here. And so what we're going to do is we're going to, again, run the simulation. And we'll see that the neurons that are slightly more excitable, the brightly colored neurons, are initially the ones that are taking care of tracking the input signal. Now, if we start inhibiting all of these neurons, we're inhibiting all the active neurons in the circuits, then what you can see is that all the brightly colored neurons are now in the back, and the dark colored neurons, they're the ones that are now in the front. And they will immediately take over the coding of that input signal. So even though we've just silenced all the active neurons in the circuit, all these hidden neurons that were previously unseen, they are still there, and they will take care of tracking the signal. I've also told you that these systems are sensitive to excitation. So to see that, we have to see that there's a strong asymmetry between inhibiting neurons and exciting neurons. If we excite a neuron, we're going to move its threshold into the bounding box. But if we move the threshold into the bounding box, then we're drastically changing now the shape of the bounding box, which we didn't do with the previous perturbations. And this change in the bounding box here causes now an error in the tracking, okay? So now we have an error in the tracking that is sort of perceivable. The last thing I want to show you is that we can also develop a geometric understanding of excitatory-inhibitory balance inside this bounding box. So to do that, let's just follow the trajectory of the readout in one specific example, which is shown here. So this is the trajectory of the readout y. And the trajectory just happens to be that it hits the threshold of the red neuron here, and the red neuron fires a spike. And the threshold of the red neuron, just to remind you, was determined by its decoding vector d1. The direction of the threshold is determined by the decoding vector d1. And the voltage of that first neuron was given by the projection of the error x minus y onto this decoding vector d1, or the dot product. So the voltage is here moving in this direction. To better understand what the voltage of the neuron actually does, we can just plot it over time. So here's the voltage of the red neuron over time. And what you can see is that initially, when this readout moves towards the threshold, the voltage of the neuron is depolarizing until it hits threshold. Then the readout is being pushed into the bounding box. That here corresponds to a hyperpolarization of the voltage. So the spike has a hyperpolarization afterwards. And then the readout moves back, leaks out again towards 0, and that corresponds to a depolarization of the neuron again. Now let's look at a neighboring neuron. Let's look at a neighbor of this red neuron. So a neighbor of the red neuron could be this yellow neuron, for instance. Now the yellow neuron also sees a projection of the readout, a projection onto a different decoding vector, its own decoding vector, which is D2. And this is now the way the voltage of the yellow neuron evolves. So it also gets depolarized initially. It gets depolarized. Then it sees a sudden inhibitory input. That happens when the red neuron fires. And then it gets depolarized again. So there are two insights we can have by looking at it this way. So the first insight is, since this yellow neuron receives an inhibitory input when the red neuron fires, we must conclude that there's just a connection from the red neuron to the yellow neuron. That's an alternative way of thinking what's happening in the system. The other insight we may have is that actually this neuron has seen excitation rising, and then it suddenly got an inhibition that eliminated all these excitatory currents that were just coming in. So it is sort of in a state of balance. The inhibitory currents that it received just eliminated the excitatory currents. And fundamentally, the reason for this type of balance in the system is simply that all of the neurons are trying to track a coding error, okay, the input signal, but they're trying to eliminate coding errors. And that elimination of coding errors biophysically just corresponds to an elimination of errors that are excitatory currents by the spiking of the neurons, which are inhibitory currents. And that causes this type of EI balance. So fundamentally, instead of subtracting these readouts from the inputs, as we've done previously, we can also just take this argument to say, well, this is just a circuit of neurons that are laterally inhibited. And if you may wonder at this point, what circuit is this guy even simulating? Well, it turns out all we're simulating is just an integrated fire network. So all the bounding box actually is an integrated fire network just visualized in the space of readouts. And here is the equation for that network. It has a leak. So this is the voltage of a neuron. This is the leak term. There are feed-forward inputs, which are the input signals. The feed-forward weights are just given by the decoding vectors of the neurons. And it has recurrent weights. And the recurrent weights are given by dot products of the decoding vectors of different neurons. So to summarize that, I've basically showed you that this simple toy model can give us good intuitions about several phenomena. So one is why you have trial-to-trial variability in neural circuits. Another one is that you may be very robust to inhibitory perturbations, but very sensitive to excitatory perturbation. Another one is why there may be excitatory-inhibitory synaptic balance in neural circuits. And we've also seen that essentially it's just an integrated fire network with a certain amount, with a specific type of connectivity that is linked to the decoding vectors D. But now our critique can come back and say, well, that's nice, but it's still not biologically realistic. It's still just a toy model. For instance, it doesn't really compute anything, right? You just put the input and then there's the output. It also requires instantaneous synaptic transmission, and as I will show you, that is going to potentially result in seizures. It also doesn't obey Dale's law. There's no separation of excitatory and inhibitory neurons here. And adaptation, what about adaptation? We don't say anything about adaptation. Or connectivity, it's hand-designed. And what about learning? What about tuning curves? What about receptive fields? Can be a long laundry list of things that are missing because it's just a toy model. So in the last years, we've been trying to work ourselves through that laundry list to make the model more biologically realistic. So for instance, if you're interested in adaptation, there's a very beautiful paper by Gabriele Gutierrez and Sophie Deneuve, where they show how you can understand population adaptation in this framework as a trade-off between cost, the cost of coding for something, and accuracy. If you're interested in learning, we published a paper 2 years ago where we show how to learn this network with a set of synaptic spike timing-dependent plasticity rules. If you're interested in tuning curves or how you get receptive fields in this type of network, we've, for instance, shown that in a paper by David Barrett in 2016. Today, though, I want to focus on this first cluster of problems. And first, I want to explain the problem we face with synaptic delays in this system. So imagine that one of the neurons is firing here, this blue neuron. Now, when that blue neuron is firing, this light blue neuron, it's going to move the readout into the bounding box. But at the same time, it's supposed to quickly inhibit all of its neighbors. But what if that inhibition is not sufficiently fast? Well, if the inhibition is not sufficiently fast, then one of those other neurons will say, well, the error is not taken care of. I have to fire too. And for instance, this green neuron here could say, I'm going to fire too. And if this greenish neuron fires too, it's going to move the readout out on the other side of the bounding box. So these delays in communication can lead to what we call uninformed spikes. However, the problem now is that there are multiple thresholds crossed this way. Multiple thresholds on the other side of the bounding box from these reddish neurons are being crossed. So they are going to all fire to get the readout back into the bounding box. And now we have a problem, because now we get this sort of ping pong between neurons that are all going to start firing. And that leads to some type of seizure-like activity in the system, which is not good. And the biophysical cause of this is simply that there are excitatory connections between oppositely tuned neurons. So how can we eliminate this problem? Well, let me just point out that all the results that I've shown you, about EI balance, trial-to-trial variability, robustness, sensitivity, they all really just rely on the notion of a boundary in readout space. And this boundary is made up of many neurons. It's basically a boundary made of many neurons. And the problem we have with the delays and the seizures simply come about because the boundary is closed. So it's very obvious what we have to do to eliminate those problems. We just have to open the boundaries. If we open the boundary, though, what we suddenly get is a system that is all inhibitory. So all the neurons are now inhibitory. Well, this is great because there are no more seizures now, but it also has a problem because we're no longer tracking the signal. So how can we solve that? So the solution to this problem is to just think three-dimensional instead of two-dimensional, which I frankly don't really like doing. I'm more like a two-dimensional guy. And so to do that, what we're going to do is we're going to introduce a third input dimension, which is the z dimension. And we're going to say we only want to track the signal in the x and y dimension. And the z dimension, we're not going to care about. Now, the z dimension is going to just be a constant input signal that we're going to feed in order to the neurons. And then the type of bounding system we get, we call the bounding cone, because essentially it's like an ice cream cone, where the individual neurons are now the faces of this ice cream cone here. And if we do that, we can now track the input signal just as we did before. But now we won't get this problem of getting into seizures, for instance, if there's too much noise in the system. But there's also something else that we gain, which is interesting, because now we can take this third input dimension, the z signal, which is just an offset that we feed into all the neurons, and we can change it. For instance, if we increase this offset, if we increase the current we feed into all the neurons, we essentially are going to change the overall population firing rate of the system. What happens, though, is that the operating point of the system kind of changes. So it moves down, the cone moves up in the space, and the operating point moves down. And when the operating point moves down, the effective bounding box, the effective bounding circle becomes smaller. So that's a way of controlling the precision with which we encode the input signal, just by feeding in a current. Now, there are several details I'm leaving out, and because I just moved the mouse, you don't see it anymore. There was a poster yesterday from Sandra Keming and Bill Podlaski, and you can ask them about the details, or you can ask me afterwards. But let me go back to the overall notion of an inhibitory population. Because beyond the bounding cone, we can also just ask, what is it that an inhibitory population actually computes in this type of framework? And for that, we just have to see that there's a boundary here that is created by the inhibitory population. And the readout will move around this boundary. Well, to see what the inhibitory population computes, what we just have to do is we have to move out of the readout space and move into a space where we can see the inputs and the readout at the same time. And that is shown here. So here we have the input signal x, and this is the readout y. And it turns out that in that space, neurons are still, the thresholds of neurons are still just lines. And there's still a boundary, which is this black boundary. Now, the readout only moves here on the y-axis because the x-axis is the input signal. So it's only going to move down. And this way you can see that the readout is going to hover around this black line. And so this black line, which is what the readout is going to be for any input signal, that is now actually the function that this inhibitory circuit is computing. And it turns out that what this inhibitory circuit is computing is simply a convex function of x. So a piecewise linear convex function of the input x. That is what an arbitrary inhibitory circuit in our framework is computing. Now, of course, you may notice I'm talking about inhibitory population that's not even half the brain, right? A lot of stuff is missing. What's up with excitatory neurons? So to think about excitatory neurons, I want to first point out that this regime here in the inhibitory population, we're going to call this subthreshold regime, because that's the regime where all the neurons are subthreshold. And then this regime is the suprathreshold regime, where at least one of the neurons is above threshold and will fire. Now, what does the inhibitory population actually do? What do inhibitory neurons do? Well, what they do is, whenever they reach this bound, they're going to fire a spike to move other neurons subthreshold, because that's what inhibitory neurons do. They inhibit other neurons. So they move them further away from the threshold. So the effect of the inhibitory population is to move into the subthreshold regime. And from the point of view of the readout, the subthreshold regime is a convex regime, is a convex set, and the boundary looks convex to the inhibitory readout. These insights also tell us how we should think about excitatory population. If we had a purely excitatory population, it's simply going to do the opposite thing. So it also has this boundary. It also has a subthreshold regime and a suprathreshold regime. But the effect of the excitatory neurons is exactly the opposite. So the excitatory neurons are going to fire to move other neurons voltages closer to threshold, so into the suprathreshold regime, which is shown here. Last but not least, what we can do is we can put these types of populations together and that will give us an excitator-inhibitor population, where here on the x-axis, we have the excitator readout. On the y-axis, we have the inhibitory readout. Now, the suprathreshold regime is down here. The subthreshold regime is down here. The populations, the red boundary here, that's the excitatory population. So I'm no longer showing you the individual thresholds of the neurons. I'm only showing you the boundary that this population is creating. And when an excitatory spike is fired, it's going to move the excitator readout towards the right, towards the suprathreshold regime. The inhibitory population is the blue population. And when that blue population fires, it's going to move the inhibitory readout into the subthreshold regime. It's going to move the two readouts into the subthreshold regime, not just the inhibitory readout. Pardon me that. And we can simulate the system. Now you see the inhibitory readout is initially decaying, and then the readout altogether is going to move around to this fixed point here. And whenever one of the neurons fires, I'm just going to make it a little bit thicker so you see which neurons are firing. So what do we have now? So for those of you who are experts, that may look familiar because it's basically an inhibition-stabilized network. The big difference here is that these two lines, though, are not null lines. They are boundaries. They're boundaries of many, many neurons. Now, it was shown that you can actually think about this system as sort of a concave-convex optimization problem. There's a nice paper by Lee and Pelevan 2 years ago where they show that you can formalize this as a minimax optimization problem. So now we have a system that is actually quite powerful computationally. So it no longer just tracks the output, but it performs a large range of computations. And this also relates to some nice work by Robert Rosenbaum's lab, where they show how you can get nonlinear computation in balanced networks. If you start imbalancing some of the neurons, you make them more hyperpolarized. And with that, I'm going to end and just summarize what I told you. So the first thing I told you is that we already know something about spikes. The one thing that we're really good at as a field is to turn spikes into analog variables using linear methods. That we would do in basically pretty much all experimental setups. What we also know is we know how to turn analog variables back into spikes, but we usually resort to single neuron models. So what I've shown you is how to think about this problem on the population level. So to just take a population level view, not just on the decoding end of things, but also on the encoding end of things. When we solved this problem for a very specific system, which is this autoencoder, that revealed a coordinated spike encode in which neurons bound the coding errors with their voltages. This type of spike coding naturally accounts for EI balance, trial-to-trial variability, robustness, and sensitivity of neural systems, and I think it leads to some nice insights on how these things could work. Then, as a teaser, in some sense, in the last part of the talk, I've shown you that we can extend these ideas to networks that don't just track input signals, but that are able to perform computations simply by assuming that neurons create these boundaries in the readout space. And thanks a lot for listening.
Thank you, Christian. We'll move for questions. So I have a question over there, and please queue up the microphones if you have any questions.
Thank you. Very elegant viewpoint on kind of population level. I really like it. But I have what I think is kind of, I must have missed something early. Why is the readout leaking to zero? Your whole analysis seems to depend on that leak to zero. I couldn't figure out where that came from.
So mathematically, it doesn't really have to leak out to zero. You could say it doesn't leak. The reason we let it leak is twofold. So one is because usually when we filter spike trains experimentally, for instance, we always use finite kernels. So in some sense, when we actually treat spike trains, that's what we do. Another reason is that is what a postsynaptic neuron will do. It takes the spike train and filters it. And so if you want, it's not a normative reason at this point. It's more just a reason to make it match to biology.
So as a follow-up then, what have you looked at chains of these networks? So if I'm understanding what you're doing, your output neuron is essentially a single integrate and fire neuron with a leak. So at that point, have you looked at what these population view does if you go from population to population, rather than population to single neuron?
So no, we have not looked at communication between populations in this framework, basically. if that's what you're asking. So like having one population and then have another population and how they communicate, is that what you're asking?
Yeah, that's what I'm asking.
No, we have not looked at that.
Okay. So we'll take a question from the other side.
Hi, hi, great talk. I was wondering how well this scales when you have higher dimensional input. So if you want to have some hypersphere bounding box then, do you need exponentially more neurons or?
That is an excellent question. And I thought I had made some slides for this actually. Yeah, that's an excellent question. So it doesn't, so if you want to keep the bounding box like a little nice soccer ball, that doesn't scale up to higher dimensions.
Yeah.
So you have to scale in a different way, basically. And the first thing, well, let me leave this one out. But basically what happens geometrically is that if you scale linearly, many of the faces of what is mathematically actually called a polytope, but it's a bounding box, many of the faces become more and more orthogonal. Okay, that's just going to happen. So it's a much more complex object in high dimensions. But the properties I've shown you, like trial-to-trial variability, sensitivity, robustness, they actually also hold in high dimensions. But it's harder to visualize what happens in high dimensions. That is certainly true. And you cannot really imagine it to be a sphere anymore.
Yeah. Cool, thank you.
Okay, I'm going to abuse my power and ask a question. So does this have, to the right actually.
Okay.
Do you expect noise correlations?
I'm sorry I can't see it
Yeah,
because there's too much light actually.
Yeah,
That's it.
there's
It's
way too
Omri,
much
of
light,
course,
yeah.
yes.
So if this, do you expect negative correlations between similarly tuned neurons in a sense? So if I spike, I steal the spike away from my closest neighbor.
Yes, that's what you would expect, indeed.
And is
You're
this
going to steal a spike away from your closest neighbor. That's right.
so does that mean that all like signal and noise correlations will be opposite in this framework?
About signal correlation, I haven't thought, but the noise correlations between similarly tuned neurons should be negative. So you're right; they should be opposite. And so you'll say, but that's not what we observe. But then we have to think more deeply about what are we actually observing, because when we observe noise correlations, we're not necessarily just observing what I just showed you, because obviously biological experiments, it's not like high energy physics where everything is super well controlled. There's a lot of other things that are going on, that we do not control, and they may totally flood, for instance, what we see on the level of noise correlations, etc. So therefore, it's true, we should see negative noise correlations, but only in experiments that are super well controlled, that no external influences, right? The stimulus, for instance, shouldn't change around. Yeah. Okay, there shouldn't be other sort of things going on in the system that you don't have under control. Then you should be able to see it.
Okay, thank you. We have one more question.
Should be an easy question. So this is very beautiful. Where do you stand on the original question? Are realistic networks just implementation details? Are rate networks just good function approximators?
So I think rate networks are good function approximators, and there are mathematical links between this framework and rate networks. That is totally true. So you can, under certain circumstances, map them onto each other. And so you're asking me maybe a question that isn't so easy to answer, because I don't know the answer yet. Okay, I think we're just trying to push this framework more and more towards biology. And so far, all I can say is certainly has changed the way I think about neural circuits now. Very different from the way I thought about it when thinking about rate networks. And maybe that's, for me, the most important thing at this point.
Okay, so let's thank Christian again. And the next speaker is Wiktor Mlynarski from the IST Austria.
Okay, good morning, everyone. Why? Oh, that's too early. We'll just get rid of that. Okay. While I'm a theorist, I would like to tell you today about work done largely by and in collaboration with my experimentalist colleagues from the neuropathology lab at IST, which is led by Max Josch, and the work was done also by Jan Svatoň, Olga Symonova, and in particular by Divyansh Gupta, who is a graduate student in Max's lab, who has led this effort and could perhaps stand here and present this work likely better than I will. But the idea that sensory systems are adapted to the structure of natural scenes is by now very well established in the field. And the evidence in favor of it comes from many now canonical studies of individual neurons in the insect visual system, in the mammalian visual cortex, from analysis of small population of neurons, such as, for example, orientation domains in V1, or neurons which are called statistical features for high-level vision. And today I would like to add to this evidence by describing what is perhaps largest known to date sensory population, which is adapted to a single salient feature of natural visual scenes. And to do that, I'll first tell you about the retina-wide population recordings performed by my colleagues, and then I turn to the theory of efficient coding to identify 3 salient properties or features of such large populations, which will constitute theoretical predictions and explanations of phenomena observed in the data. And that's the plan of action for the next few minutes. So first, let's talk about the data, and we are talking about the retina here, the beginning of the visual brain. And I'm certain that everyone here knows that we can record activity of retinal ganglion cells in optical imaging experiments where we position a microscope above some field of view. And then if we want to record more cells, we can move that field of view, sequentially stitch number of them together, and obtain on the order of tens of hundreds of neurons per retina. But if one is interested in population-level phenomena, this approach is insufficient. And to address this issue, Divyansh and Max developed a new approach which allows one to record up to thousands of neurons within a single field of view. And such a single field of view can cover up to 40% of an individual retina. Moreover, Divyansh was able to cross-register neurons from many different retinas in the same coordinate system and create this fantastic data set of almost 27,000 neurons from an entire retinal surface surrounding the optical nerve here in the middle. And for each of those cells, we know not only its position, of course, but we can also estimate their receptive fields. We can do that using the canonical standard white noise checkers, and this gives us somewhat crude receptive field estimates. So in order to improve this quality, we could, for example, use higher resolution, higher spatial frequency random checkers, and that results in noisier estimates. But in order to alleviate this issue, my colleagues developed a new class of stimuli, which are not only flickering, but also moving spatially. And that results in these highly structured and robust estimates of receptive fields. And unbeknownst to us, in parallel, this approach was developed and published recently by Pamplona and colleagues. So now we can estimate tuning properties of neurons in the entire population. And for each individual neuron, we know not only its position, but also, or receptive field, sorry, but also its position in the retina, in the consequence within the visual field. So now we have this complex data set of tens of thousands of receptive fields and knowing their positions in the visual field. And we would like to find some low-dimensional structure in this data, which is not only interpretable, but could be understood as manifestation of some design principle. And here I'll turn to the established theory of efficient coding of natural visual scenes. Because if one lives one life as a mouse, one perhaps encounters some properties of one's environment which are salient enough and could be exploited by entire retinal population to increase coding efficiency. And one such property is for sure spatial distribution of power along elevation azimuth, which we computed by averaging images from the upper and lower visual fields in the UV spectrum. So there is this characteristic pattern here which emerges, right? At the top, where it's brighter towards the sun, right? The power of the stimulus is stronger, and then it decays towards the ground with this characteristic transition at the horizon line. And we found this out using data kindly shared with us by Luca Abale and Hiroki Asari, but similar results were also recently reported by Yongrong Q and colleagues from Thomas Euler's lab. So now, in order to understand how retina might exploit this property of natural scenes, I need to develop an encoding model or coding model of retinal ganglion cells. And we assume that stimuli here originate from somewhere within the visual field, are first represented by noisy responses of photoreceptor cells, which are then pulled by retinal ganglion cells characterized by some mysterious receptive field W, which then generates response R, which is conveyed down to the brain. And we'll assume here the simplest possible model of the response, linear in the receptive field and the stimulus, which can be trivially decomposed into a difference between the outputs of the positive components of the receptive fields and negative components of the receptive fields. Now, we'll try to find such receptive fields which minimize, on average, the total amount of activity in response to a large set of natural images in order to minimize use of resources. Now, when this activity will be minimized? Well, it will be minimized when this positive component and negative component cancel each other out. In some sense, predict each other, and what's conveyed down to the brain is the prediction error, right? Hence the term predictive coding. We also added here a weak prior to which prioritizes pooling of localized photoreceptors, and what emerges from such optimization problem are receptive fields with this characteristic center-surround shape. Now, this class of models is famously very sensitive to signal-to-noise ratio. In our case, power of the noise is assumed to be uniform across the visual field because it's inherited from properties of photoreceptors. But power of signal, of course, depends on the position. So here at the top, power of the stimulus is high, and noise fluctuations are relatively weak, and SNR is large. But if you go downwards, the power of the stimulus decreases, noise starts to dominate. So this means that this vertical axis of the visual field spans basically a dynamic range from low to high SNR. And the model predicts different receptive fields for different values of signal-to-noise ratio. So here at the top, where SNR is high, we expect receptive fields with relatively small center and quite sharp surround. And with decreasing SNR, the center will be slightly increasing, while surround will become more diffuse. And I think it's better visible if I plot cross-sections of the receptive fields. And the reason for it is because one way to think about it is those two components, center and surround, have to, in order to cancel each other out, they have to average over spatially larger areas to cancel out the effect of increasing noise. So now a prediction starts to emerge, right? Because if we take this vertical dimension, we'll expect there to be an increase of the surround strength along it, and at the same time decrease of the center size. We can also form the third prediction when we look here at the horizon line, where stimulus has this characteristic asymmetric pattern, and the model predicts that the optimal receptive fields would exploit that structure by pulling asymmetry with an asymmetric surround, which means that some measure of surround asymmetry should peak somewhere close to the visual horizon. And these are our three predictions, which we derived from that very simple model derived from a general theory, but they will be robustly predicted also by a number of efficient coding approaches, which share one largest common denominator, which is transmission of sensory information in presence of strong resource constraints and noise. So now our theory helped us to identify three features of this large population: vertical trends of the surround strength and of center size, as well as a peak of surround asymmetry at the horizon. And the only thing left is to verify whether that holds empirically. So first, let's have a look at the surround strength, which should be increasing with the elevation. And I plot a map of it here. And it indeed does. It's higher at the top than at the bottom, so more purple at the top, more green at the bottom. And I plot it as a function just of elevation. It shows this characteristic shape which resembles the theoretical prediction here at the top. And that holds not only on average, but also across individual retinas. Center size shows the opposite trend. It decays with the elevation within the visual field, in agreement with the theory. And finally, this surround asymmetry forms this kind of streak, or in the elevation domain, a peak close to the horizon, also in agreement with predictions of efficient coding. So to summarize this agreement of theoretical predictions and empirical reality, I can plot the predictions of optimal receptive fields for three different elevations and corresponding averages, or a map of receptive fields within a single retina. So altogether, this seems to imply that retina does indeed seems to be holistically adapted to this panoramic structure of natural visual fields. And I could then here But for the remaining, like, say, 30 seconds, I would like to tell you a micro story about our experience with theory in that project. Because it wasn't at all this kind of theory, prediction, verification sequence. What happened chronologically first is that Divyansh and Max observed this kind of very strong salient streak of asymmetric receptive fields at the horizon. And we've been discussing what might be the reason for it, and realized that perhaps it could be manifestation of efficient coding. But then when we committed to a theory, it immediately generated predictions about vertical trends of receptive field properties. And importantly, if it wasn't for this well-established theory, we would have never looked for these properties of the data, because while systematic and significant, they are quite subtle. So I think this experience nicely illustrates different roles of theory, such as explanation or prediction, and how they can interact in neuroscience. So with that, the only thing to do is to thank again my collaborators, Mark, Jan, Olga, and in particular Divyansh, who is here at Cosyne. Just has waiting there, just has longer hair and more beard now. So if you see him, please ask him all the hard questions. And I would also like to say thank you to Gaspard Kacik and colleagues from Theoretical Biophysics and Neuroscience Group at IST. And I deleted the thank you slide, so thank you very much.
Thanks, Wiktor, for a beautiful talk, and you can line up to the mics. In the meantime, I'll ask a question myself. So, do you think this is something that is developmental or innate? So, do you expect this pattern to be due to visual experience, or does it is it genetically encoded?
Yes. I think that this specific pattern, the way we think about it right now, or rather I think this specific pattern is more, is due to visual experience over evolutionary timescales. So I think that it would be present already in the newborn retina, and we could ask Divyansh how old are the animals where this experiment was performed. But perhaps there are effects which occur on top of that, right? This very salient property. Yeah.
Okay, thank you. Question from there.
Hi. Yeah, I was just thinking about the starting idea that signal to noise increases going up from the horizon, and that's just based on the idea that the mean luminance increases going up from the horizon.
And contrast, so basically square power of intensity, yes.
Okay, good, so I didn't get that. Thanks very much.
Thank you.
Hi, very nice talk. The diversity of these responses are all from the same subpopulation of retinal ganglion cells, or do you think that these are morphologically or genetically distinct retinal ganglion cell subpopulations?
Yes. So I for sure won't do justice to the nuance of this issue, but I think in my understanding, these are, we can think of, this phenomenon being present across all types of retinal ganglion cells in the retina. And actually, if you look at our preprint, we did some simple functional clustering to functional subtypes, and we see that this trend holds across different functional subtypes.
Okay. Thank you.
Thank you.
Okay, so let's thank Wiktor again. And move on to the third speaker this morning, Madhura Ketkar from the University of Mainz and the Institute of Developmental Biology and Neurobiology.
Okay, so hi everyone. Throughout the day, we come across many different light levels, but we don't want the changing availability of light to change our perception. In other words, we want luminance invariance. And this is what I'm going to tell you today, how flies achieve luminance invariance, especially when the changing environments make it challenging to do so. So, perceptual invariance across changing illumination is a part of our day-to-day experiences. Let's imagine playing soccer. you may play in bright daylight or in twilight, but the typical soccer ball you will always perceive as a combination of black and white patches. Let me zoom in on the soccer ball a little bit. We have again two scenarios, one dim, one bright, and there is always a very big contrast between the black objects on the white background, but what reaches our eyes is the mere luminance difference, and this is going to depend on the availability of light. But what our brain does to make the same sense of these two scenarios is decreasing the gain when transitioning from 1 to 2, or increasing the gain when the transition is the other way around. So this is a two-way gain control which ultimately achieves normalization of the mere light difference such that it computes a luminance invariant measure of this difference, in this case Weber contrast. So a lot of this gain control is already happening within photoreceptors, the first ones to encounter the difference in light, but the photoreceptor adaptation, which is responsible for gain control, is not instantaneous. This means in dynamic conditions, adaptation alone may not be able to reach invariance per se. Let me illustrate how. Let's pretend that the two scenarios we have been seeing so far are merely representing two different times of the day. Then there is sufficient time for adaptation, and the adaptation driven response is then expected to be similar, which means it is expected to be invariant to the light levels. But if we are rather playing in dynamic conditions, which we are always playing, if the ball suddenly goes into a shade, the difference in light is now evaluated from an adaptation state which is in a bright context that was existing before, and therefore the difference is perceived to be smaller than it actually is. Vice versa is also true. So when the ball suddenly comes out of a shadow, the same difference will be perceived really bright because we are adapted to the dim conditions that were there before. So this is not how our perception is working. Our perception is rather driven by the contrast driven responses, and contrast remains the same throughout all four scenarios, and therefore our perception also remains the same. So the question is, how in these dynamic conditions are we achieving invariance past photoreceptor adaptation? We started asking this question in the fruit flies, Drosophila melanogaster, and the advantage we have here is we can specifically manipulate single circuit components in this visual system, and we can also combine the readouts here with modeling approaches to gain a deeper insight into how the perceptual invariance is achieved in dynamic conditions. So by theory, you can imagine that we need access to luminance information to further control luminance gain downstream of photoreceptors, and this is what we looked for first. So in the fly visual system, downstream of photoreceptors, we have the so-called lamina neurons. I am showing you two such neurons here, and we first explored if they have properties which can encode luminance information. L2 neurons, for example, only respond to changes. As you can see, they have a spike-like response only when there is a change in the stimulus. So there is no retained information in these neurons, only the information about change. In contrast, L3 neurons have sustained response properties, and these sustained components retain information about luminance. So if we quantify these, we actually get a reliable correlation with the luminance information. So this could be the information that we need for adjusting gain further downstream of photoreceptors. So we tested this hypothesis by asking flies now instead of single neurons, and for this purpose we recorded responses of the flies when they walked on a ball and responded to the visual stimuli we were showing to them. And we did this in two types of flies, one the usual wild type flies, and the other set where we particularly silenced the L3 neurons, so the luminance information would be gone in those flies using genetic tools. And this is what we observed. So to start with, the wild type flies responded not much different when different stimuli were shown of the same contrast but changing luminance. So we mimicked the dynamic conditions here. There was invariance in wild type responses, but the L3 silenced flies instead underestimated the dim stimuli selectively. So here we are basically recapitulating the scenario where a sudden dim condition is encountered, and then there is no luminance invariance anymore in these flies. And this was not true only in this particular set of experiment, but when I repeated this experiment in different absolute luminance cases. For example, I used neutral density filters and repeated the experiment. In each set of experiment, I saw that there was deficit, particularly in the contextual dim light. So we have one answer so far, that L3, the neurons that encode luminance information, are necessary to adjust gain past photoreceptors to get invariance in suddenly encountered dim conditions. But what about suddenly encountered bright conditions? In principle, they were also present in the experiment I was doing, but we didn't see a case where L3 silence flies overestimated the stimuli. So we hypothesized that so far we were using 100% contrast, which means the biggest contrast the flies could see, and therefore there was perhaps response saturation. So this time we chose 2 smaller contrasts. You see that for each of these cases where we did basically the same paradigm, kept contrast constant and changed luminances, we have overlapping traces, which means we again have invariance in wild type flies. But when we silence L3 neurons, the first thing you notice is that the traces are not overlapping anymore, so there is no invariance, plus some of the response traces are higher than the wild type flies, meaning there was now overestimation of responses in bright conditions. If I quantify this, you will see this even clearer, plus you will see that actually the overestimation on silencing L3 is present in all different luminance cases. So now we have seen 2 cases mimicking scenarios 3 and 4, where again the L3 neurons having luminance information are necessary to adjust the gain in 2 different ways to achieve invariance. But we are still seeing this in 2 discrete cases of discrete contrast. And this somehow didn't make sense to me. For example, we are looking at luminance gain control, and why does it have to be dependent on the contrast value? So what we did further is again testing this minus 50% contrast at different absolute luminance cases. So we repeated something we had done previously with 100% contrast, and these are the results I am showing to you. You see that in dimmer regimes, dimmer absolute luminance regimes, there is actually underestimation of this 50% contrast, whereas in the brighter regimes there is overestimation. So far we were, I had shown you only one of the experiments. Now I repeat that experiments at 5 different luminance regimes, and we actually see both under and overestimation of responses. Furthermore, we can actually recapitulate this kind of 2-way deficit across absolute luminance if we simply model the L3 silence responses as a linear function of the contrast only responses, or the change only responses that I had shown you with L2 neurons. So we do see a 2-way gain deficit as we were expecting in the beginning, but now I would like to show you that this deficit exists both at absolute as well as contextual luminance scales. So I took the same data set. This time I am choosing the analogous stimuli in all 5 sets of experiments, and I am going to draw a line through them. The line is more or less horizontal across a large range of luminances, meaning there was invariance in wild type flies. If I do the same with L3 silence responses, I don't get the straight line. This also we have seen that it's not invariant anymore, but let me remind you that this is not contextual, this is not sudden changes in luminances anymore. We are looking at 5 different experiments happening at different time points as if they were happening at different times of the day. So we have a deficit in adaptation adjusted gain that exists at the absolute luminance as well, or at the slow time scales of changes as well. There is the deficit is two-way again. On the lower side, it leads to underestimation of responses. On the higher side of this crossing point, it leads to overestimation of the responses. But furthermore, if I draw the lines showing the observed L3 silence response, these lines are again crossing the orange line in the middle, which means there is again a two-way gain deficit, which now exists at the fast time scales, because now all the stimuli were shown on changing on fast time scales. So there is a coexistence of two types of deficits, one at fast and one at slow time scales. And the same L3 neurons with their luminance information are correcting for this. So our next question was, how can this happen in the circuitry? The same neuron is responsible. So we needed to understand algorithmically how these different gain corrections can take place in the circuitry. For this purpose, we started with the observation that there is a clear crossing point in this data set, and this is where our collaborators, Shuai, a graduate student from Julijana Gjorgjieva's lab, came into picture. He started with our basic model, which could only predict the L3 silence responses based on a linear model of L2 responses, the contrast only responses, and he proceeded with adding more information to this model. The first thing Shuai did is making a function of luminance, which can now tell the model if we are on the left or the right hand side of this crossing point. When we are on the right hand side, this term compensates for the contextual deficits in the responses. So now we expect that with addition of this model, we come from the green lines to the orange lines. But we still have to go down to the black line, right? So then Shuai added another term where if we are on the left side of the crossing point, this term now only boosts the responses based on the luminance information alone. With this, we do expect to correct for the deficit in absolute luminance and come down to the black line. So let's see how the model is performing. This is the model now on the left hand side, and on the right hand side is the data. You can compare and see that we were already able to predict L3 silence responses well just with the L2 neuron responses. But now we also get luminance invariant responses for wild type flies, and this happens for both the contrast sets. However, you must have also immediately noticed that we still have some discrepancy between the model and the data, that we don't have the overestimation of contrast we see with 50% contrast, for example. So we still need to add an inhibitory component to this model, and this is what we explored next. What kind of stimulus parameter can provide us the reliable information for this? So if we looked closer to the stimuli that we have used, we immediately see that there is a difference between 50 and 100% contrast, that the 100% contrast always had a moving edge that was fully dark. So it always had completely zero luminance, whereas the 50% stimuli had edge luminance that also varied with the background luminance to keep up with the 50% contrast. So we thought that if we base the inhibition on the edge luminance, we will exactly get the required illumination for 50%, but it won't come into the picture when we are predicting responses for the 100% contrast. And for this purpose, Shuai added another term based on the inhibition, and now the model actually does predict the overestimation of contrast that we have seen for lower contrast and higher luminances. So now the model, the complete model, predicts all different aspects of our data well, namely the luminance invariance in wild type flies, and also both under and overestimation that we have seen at different luminance statistics. So I would like to conclude now. First, I started showing you that there is a problem with dynamic conditions if we only rely on photoreceptor adaptation. And then I showed you that luminance information, which we found in the L3 neurons, is required for gain correction, which happens past photoreceptors to achieve luminance invariant responses that we would aim for. Later, I proceeded to show you that even at slow changes, even at the time scales where the changes are because the sun rises up or down, photoreceptor adaptation is not sufficient to achieve invariance. Instead, we also found over and underestimation, the two-way gain deficit at slow time scales, and the same luminance information in L3 was responsible or was necessary to correct for this deficit by implementing further gain control. So with that message, I would like to thank our labs. So it's a collaborative project between the two of our labs. I would like to thank Marion and Julijana for their excellent supervision, and the lab members I have listed here, or not listed here, but shown here, who have directly or indirectly contributed to this project. I would further like to thank Carlotta Martelli, Viola Prizeman, and Jan Benda for their advices that have improved our understanding of this project a lot. Finally, I would like to thank our host institutions as well as funding agencies, and also thanks to Cosyne for this wonderful opportunity. So thank you, and I would like to answer your questions now.
We have time maybe for one quick question. Okay, so maybe I'll just ask one clarifying thing. So the wild type responses, I'm not sure if it's just noise, but they seemed all to have a bit of a negative slope, and that model didn't. Is that something that's just my illusion, or?
I just go back to that slide.
Right, so the gray ones, let's say on the top, on the bottom right, all have
this
tiny
slope you mean?
negative slope.
Yeah, we were also puzzled by this. We don't think it is actually related to how much they perceive the stimuli, but rather there is a component of startle which is present, which is, I think, proportional to the brightness of the stimulus. So it exists in the beginning, but later the amplitudes seem to catch up the same turning velocity. Okay. Just because I'm showing you the total turning here, I think we see a bit of negative slope, but not related to the computation itself.
Okay. So thank Madhura again.
Thank you.
And now for the last speaker, Francesco Randi from Princeton will take us to see elegans.
All right. Hello, everybody. Let me see if I have a pointer. So my name is Francesco, and I'm a postdoc in Andy Leifer's lab at Princeton. And today I want to talk about our direct measurements of functional connectivity in the brain of C. elegans. C. elegans is a tiny worm, and it's a powerful system to do whole brain studies at cellular resolution, not only because it has a very tiny brain of only 300 neurons, but also because we have other data sets available, like, for example, whole brain, just single cell gene expression data. So our project starts from the question of how we can experimentally characterize the equations that govern the neural activity in a network of neurons, and in our case, the C. elegans brain. So whatever approach you want to take, ultimately you want to measure the causal interactions between neurons. And so this includes information like the strength, the sign, the causal direction, but also the temporal properties and the variability of the connections. And if you have this information is important if you want to understand how high-level properties of the brain emerge from the underlying microscopic circuits, but also if you want to better understand the relation between structure and function in neural circuits. So let me point out, in this talk, I'm going to talk about functional connectivity, referring to these properties. And I want to stress that this is the causal functional connectivity and not just the correlation of neural activities. So C. elegans famously has its anatomical connectome mapped out. And this is a map that tells you which neurons connect to which. But if you want to think about or simulate neural dynamics, the connectome itself lacks some important information like, for example, the strength and the sign of the connections. And so a direct experimental approach to measure these causal interactions, functional connectivity, is to take your network, perturb a neuron, and measure how the other neurons respond to this perturbation. And to do this, you need to do simultaneous optogenetics and neural activity imaging. There's been several works combining optogenetics and imaging, and anyone who wants to do cell-resolved optogenetics and population imaging has to deal with certain challenges. And so I'll briefly talk about our approach that we're using as our working solution in the worm. So first of all, you need to be able to target each neuron independently and at the same time do population imaging, or in our case, whole brain imaging. And so we built this microscope that allows us to target individual neurons using a 2-photon temporally focused spot that is roughly the size of a neuron, and then that we can position arbitrarily in the sample volume. And at the same time, we do whole brain calcium imaging using a spinning disk confocal microscope in single photon. So the second challenge is that you need to have an activity indicator and an optogenetic actuator that are spectrally compatible, which means that you can activate or you can image the activity indicator without simultaneously activating also the optogenetic actuator. For this, our worms have, and this is particularly important if you do single photon imaging. For this, our worms express in all the cells GChem6S, the calcium indicator, and the optogenetic system GIR3 PRDX2. Which is sensitive to purple light. And this GIR3 and PRDX2 also help us with a related challenge, which is the toxicity of these molecules when they're expressed paneuronally in the worm. GIR3 and PRDX2 are proteins that are native to the worm. They're expressed in the worm already in a few neurons. We took them and expressed them in all the neurons. So they're relatively well tolerated by the worm. But to further reduce toxicity, we also use the QFGR system for drug-inducible expression. Finally, you also want to take advantage of the fact that worms have stereotyped brains, and so each neuron has its own identity, but it's difficult to assign these identities in fluorescence images. And this challenge has been recently solved by the community using the neuropulse strain, which is basically a deterministic rainbow for the worm. And so our microscope has also a multi-channel imaging branch that allows us to acquire this kind of images, where each neuron has, well, a different position, but also a different color. And by comparing these images with an atlas, we can assign identities to these neurons. Once we take these images, our experiments are whole brain imaging sessions in constrained worms that last more or less 45 minutes. And here you have calcium traces with the neural identities here on the left. And every 30 seconds, we deliver a half a second optogenetic stimulations to cells that are randomly selected and that are expressed here at the top. And so, for example, we can take the second stimulation here. We stimulate neuron I3, and then we can extract data for 2 neurons, for example, the left and right M3 neurons. And we see that the M3 neurons respond to stimulations of I3. And this response actually corresponds to a connection in the anatomical connectome. This is a single trial, but thanks to the neural identities, we can also aggregate data across trials and animals, and we see that this response is stereotyped. We can not only see responses for direct connections, we can also see responses mediated over indirect connections in the brain. And for example, here we stimulate neuron M3, and we see a response in MI, which receives direct input, but also in neurons I3, I4, and M1, which received indirect inputs through MI. We can do this for all the pairs of neurons that we, all the neurons that we stimulate and all the neurons that we observe. And we can build this functional connectivity map, which was one of the main goals of our project. And in this map, the example of I3 to M3 is 1 pixel here. And this map currently aggregates data from 44 animals and covers 90 neuron classes with their own identity, and stems from roughly order 1,000 stimulation and whole brain response events. And this is a map that roughly shows the strength and the sign of the connection. It shows the amplitude of the responses. So if we take our data set and look at the likelihood that in our data set neurons are connected, we can plot this likelihood against the average number of synaptic contacts in the anatomical connectome, and we see that at least in C. elegans, the anatomical connectome is not sufficient to predict our data. And this is consistent with the same type of analysis performed using just the correlation of the activities in the neurons. And in a couple of slides, I'm going to show you what we think is at least one of the reasons why this is happening. But before that, we're also doing analysis that aim at extracting out the specific signals that are propagating in the network during our experiments, and that we can use to characterize the connections independently of those signals. And to do this, we fit kernels that give you the activity of the downstream neuron when you convolve them with the activity of the neuron that we targeted optogenetically. And to fit these kernels, we parameterize them as sums of convolutions of decaying exponentials, which is a flexible way to describe causal interactions. And these kernels help us also unveil, let's say, different types of interaction. So for example, here on the left, we have an interaction that is roughly stereotyped. The kernels from different trials all are very similar. And this is the connection from AVJ to IL-1V. These are two neurons in C. elegans. And here on the right, instead, we have a connection that we might want to call bimodal. It's variable. And this is the connection from AVD to ASG. And here we have kernels fall into two classes. The orange and the green curve here are faster kernels, while the blue and the red one are smaller. And this bimodality might hint to underlying different network states, let's say. So while these kernels are the actual response function, only if you take some linear approximation, they are always well-defined, and they can be computed also from models of nonlinear networks. For example, starting from non-equilibrium Green's functions, for which we have a paper out. All right, so let me show you two examples of connections that are different from what you would expect from the anatomical connectome, and they're different in two ways. So let's take the well-characterized example of AFD to AIY. These are, again, two neurons in C. elegans. If you take the connectome and you look at the expression for neurotransmitters and receptors, you would infer that there's an inhibitory connection between a chemical synapse between them. But we also know that there is an excitatory extrasynaptic connection between them. And so if you don't measure functional connectivity directly, you're left with the question of how do these two opposing pathways combine. And you can resolve this by measuring functional connectivity. And this was one of the very few connections that was already characterized in the literature using optogenetics and electrophysiology. And people have shown that the connection is excitatory and is also close to linear. And we use this well-characterized example to validate our all-optical experiment. So this is an example in which extrasynaptic communication plays an important role. And recent gene expression data from the Sengen project hints towards an even broader role for extrasynaptic communication that goes on connections that are beyond what it's reported in the anatomical connectome. In fact, they show that many neurons express neuropeptides that are involved in extrasynaptic or wireless communication, and they also express the receptors for these neuropeptides. To study this in more detail, we consider the role of neuron RID, which outputs signal almost exclusively extrasynaptically or wirelessly. And we looked at the expression data for receptors to transmitters that are released by RID. And we compiled a list of neurons that we think receive direct inputs from RID. And so we see many of these neurons respond when we stimulate RID. And we can also show that several of them are actually several of these responses are actually mediated extrasynaptically because we can take mutant worms, UNC31 worms that are defective for extrasynaptic communication, and we see that the responses that we observe in the wild type are weaker or less frequent. We can also take the maximum amplitude of the responses and plot them against the expression level of these receptors that receive inputs that sense the transmitter released by RID. And we see that the bare expression levels are not sufficient to describe the amplitude of the responses. There must be some other factor. So this, I also want to stress that, so in our experiment, we just stimulate the neuron and measure the responses of the others. And so while extrasynaptic communication is usually thought about in the context of neuromodulation, so modulation of excitability of neuron or of our other connections, here in these experiments, we only detect when extrasynaptic communication elicits directly a response. And I hope these two examples also highlighted why it's important. Doing these characterization in C. elegans is important because in C. elegans we have the potential for complete measurements where we don't have hidden or unobserved variables. And C. elegans, these characterizations in C. elegans can teach us recipes about how to combine or how to interpret these different data sets. All right, so let me conclude by summarizing. I've shown you what, to our knowledge, is the most comprehensive causal functional connectivity map in C. elegans, and we measure this directly and using perturbations and not from correlation of activities. And I've shown you a few examples of good agreements of our data with either prior functional measurements or wiring in specific circuits. And I've also shown you that our measurements are able to capture extrasynaptic or wireless connection. And okay, so with this, I just want to thank you for your attention and also the other people involved in the project. Thanks.
Thank you very much. And we have a question here.
Hello. Very good talk. So, two questions. You mentioned 44 animals or 40 animals. So, when you do these experiments, do you see any differences with respect to animal? The reason why I'm asking is that most probably you immobilize the C. elegans, right?
Yeah, exactly. Yeah.
Yeah. So, in that context, maybe you can comment on what happens if the animals are in a different state or if they're moving or not moving. So.
Well, so you're correct. We immobilize the animals because otherwise it's technically very challenging to stimulate neurons and track them as the animal moves. We do not have a specific control on the state of the animals. What we see, the effect of different states is visible in variability in the responses that we observe. And I've shown you that characterization with the kernels, and we're working on good ways to report this variability because not all the variability is captured, for example, by a simple correlation between the responses.
Okay, a question from the other side.
Do you have any ideas about how to model the kind of extrasynaptic communication that you talked about in some kind of network model?
I mean, so because as I said, because we only measure when we see effects of extrasynaptic communication only when it elicits directly responses, I think it would be just another, I don't know, edge in your network graph. So yeah, that would be my answer. Yeah. Does that answer the question? Okay.
One last question, and then it's time.
Hi, I really liked your talk. I'm curious about how your work compares with the work of Pamela Douglas in Vienna, where they did a lot of work looking at C. elegans in a particular state, let's say moving slowly forward or sleeping, but they couldn't, so they're not stimulating one neuron, but they can see some of the connectome's activity. And so what I'm wondering is, could you use your method for C. elegans in a particular state and get the whole connectivity, the whole activity of the connectome?
Okay, so, well, the answer to, I think, your last part of the question is yes, we potentially can take C. elegans in specific states. We currently don't have, we currently, in our instrument, we don't have ways to control for that, but we could add that. But although for how the instrument works now, we don't have the capabilities yet to do this in moving animals. And so the forward-moving animal, that will be more difficult. So regarding the connectome, so we also have examples in which our responses match the connectome, but this result in which the responses or the activities do not line up exactly with the anatomical connectome, this is also consistent with the type of activity that, for example, the Zimmer lab observes. So if you look at that and you assign identities to the neurons, and you look at you plot the correlation of the activities against the same x-axis, the average synaptic counts in the connectome, you also see something that has a very small correlation. So for specific circuits, yes, we observe some stuff that is similar to a connection. In general, at brain scale, there's other factors contributing to these responses.
Thanks.
Okay, so let's thank Francesco again. And we will meet here in 20 minutes.
All right. So our next speaker is Arsene Finkelstein, who is coming to us from Janelia. And he's going to talk about optogenetic mapping of circuit connectivity in the motor cortex during goal-directed behavior.
Hi, everyone. So my name is Arsene, and today I'll talk about my work in Carl Swoboda lab that I did during my postdoc there, about optogenetic mapping of circuit connectivity in the motor cortex. And this talk will rhyme with the talk on functional connectomic measurements in C. elegans that we heard in the previous session. So before I start, I would like to thank Carl and my collaborators on this project, Kevin Deye and Randal Shan. So brain functions are believed to be an emergent property of connectivity between neurons. And there is a lot of effort to study structural connectivity at the level of individual synaptic connections using electron microscopy, like the flight connectomes that you see, on your left. So while structural connections represent the scaffold that is required for neural interactions, it is hard to link structural connectivity to function for two main reasons. So first, it is hard to measure structural connectivity routinely and combine it with functional measurements of neural activity. And second, the actual effect that one neuron can exert on another neuron in a living brain would greatly depend on the activity of other neurons in the network at any given point in time. So for example, neuron A can activate neuron B either directly or indirectly via an intermediate neuron C, and these functional connections can be modulated in different behavioral contexts by another neuron D. And I'll refer to any form of causal functional interactions between neurons as effective connections. And because effective connectivity provides information about strengths of functional interactions between neurons, studying it may reveal potential network motifs that underlie dynamics and computation of neural circuits. And today I'll describe a new method that we developed to measure the pattern of effective connectivity in a living brain. And then I'll show a link between effective connectivity and the functional properties of neurons in the mouse motor cortex during behavior. So to study effective connections, we developed an optical method that allows a rapid mapping of interactions between neurons in vivo. In this method, we are activating an individual neuron by shining light on it, and then measure if some other neurons also respond to the activation of this target neuron. So if we activate neuron A and see response in neuron B, it means that there is an effective connection between these two neurons, and I should stress that this connection does not necessarily have to be monosynaptic. So from a technical standpoint, what we do is we perform two-photon stimulation of individual excitatory neurons expressing soma-targeted opsin, and at the same time we are imaging the neural activity of the other neurons in the field of view, and using activity indicators GCAM6, we only stimulate and image from excitatory neurons. And this allows us to detect evoked responses in non-stimulated neurons. So by doing volumetric imaging of neurons located at different depths and randomizing the stimulation patterns, we can map effective connectivity between about 500,000 of pairs of neurons in about 30 minutes. And this represents almost a tenfold increase over existing methods in terms of number of potential connections that we can probe in a given time. So this is an example of how this experiment actually looks like, and the effective connectivity map that we can generate from a single mapping session, in which we probed about 500,000 of possible connection pairs. And the strengths of the connections are indicated in red, and the dot size indicates the number of output connections of different neurons. And I'm only showing excitatory connections here. Most of the strong connections tended to be between nearby neurons, which is consistent with previous findings in brain slices and in anatomical reconstructions. And using this method, we can now begin to study the pattern of local connectivity, local neural interactions, and relate to this pattern to functional properties of neurons during behavior. Movement, since movement is a hallmark of behavior, we focused on measuring effective connectivity in the motor cortex in the context of action selection. And the neural mechanisms of action selections were often historically studied in animals that went through an extensive training to perform a specific behavioral task, which might alter some aspects of local connectivity. And instead, we wanted to develop a task to measure effective connectivity before it is potentially altered by training. So we came with a new behavior, a very simple behavior that doesn't require any pre-training and yet allows to reveal the functional properties of neurons in the motor cortex. And this behavior simply requires the animal to lick a target for water reward. The target can appear in different horizontal or vertical positions around the mouse face, and the animal simply has to reach it with its tongue to obtain the reward. And this behavior is something that the mice do immediately from the very first session, and it's compatible with head fixation, which is required for connectivity mapping experiments. So to characterize neural activity in this new behavior, we used two-photon calcium imaging to record the activity of thousands of neurons in the motor cortex. And we observed that many neurons, like these neurons that you see here, responded when the water was presented in a particular position and around the mouse face, but not in other positions. So in fact, from the very first day of behavior, we found that the large fractions of neurons in the motor cortex had very prominent tuning to the target position. So here, what you see is a response of one example neuron when the target is presented at different positions around the mouse face, and different plots correspond to a trialed average activity of this neuron at different possible target position. And this neuron is active when the animal licks the target presented at the bottom left part of the field, demonstrating directional tuning. When we look at how neurons with different directional tuning are organized in an anatomical space, we did that by conducting volumetric calcium imaging across large parts of the motor cortex. Here I'm showing you a collapsed view across multiple imaging planes. We saw that individual neurons with directional tuning are actually scattered in a kind of salt and like pepper, salt and pepper like distribution across an anatomical space. And what you see is the different dots are individual neurons color-coded by the directional tuning. Neurons without directional tuning are shown in gray. And as I said, there is no apparent structure across large anatomical distances in the motor cortex. But if we look more closely, we do see that some neurons have clustering with, and these clusters seem to follow the directional tuning. So we see that we see a cluster of blue neurons over here, green neurons over there, and so on. And when we quantified it, we indeed found that neurons that were close to each other tended to have similar directional tuning. And such similarity in tuning properties can arise from local interactions between neurons, as suggested by many theories. So now we can combine behavioral experiments and functional mapping of the neuronal responses with effective connectivity mapping to begin studying this relationship. So first, we measured whether neurons with similar directional properties are preferentially connected. We indeed found a like-to-like connectivity among directionally tuned neurons in the motor cortex. So neurons with similar tuning tended to excite each other, and neurons with opposite tuning tended to inhibit each other. When we look at the connectivity profile between neurons as a function of anatomical distance, we saw the following. So we can measure the lateral distance between neurons, meaning the distance in x, y, and axial distance, meaning the distance in z. And we found that neurons that are positioned close to each other, mainly one above the other, tended to have strong excitatory connections, which is shown here in red. And the connection strengths quickly decayed with lateral distance. This connectivity profile may suggest the existence of functional mini-columns in the mouse motor cortex. Also, the like-to-like connectivity in directional tuning was apparent only for neurons residing within this functional mini-column at less than 100 microns away from each other. And we know that columnar organization of the neocortex is, of course, a hallmark of the primate brain, especially in sensory areas. And our effective connectivity mapping experiments may reveal the existence of functional mini-columns in the mouse motor cortex. We also observed that stimulated neurons tended to inhibit neurons residing further away at about 100 to 200 microns laterally, with the inhibition shown here in blue. And overall, the excitation inhibition profile resembled that of a Mexican hat, although the inhibitory responses that you see here that we can detect with these methods are very tiny. So far I described only a coarse relationship between effective connectivity and functional properties. However, these maps are pretty complex. We found that pattern of local connectivity is actually highly not random. And there are neurons that have many more effective output connections than other neurons. So we will refer to these neurons with large number of connections as hub neurons. And I should emphasize that this hub neurons seems to be a part of a continuum rather than a discrete population. And network hubs were shown to be important in many biological and artificial networks. And despite the brain being maybe one of the most well-studied biological networks, we actually don't know if there are neurons that function as hubs in the living brain. And that's because measuring connectivity is very challenging. And so far, there were only a handful of studies that attempted to measure the heterogeneity of local connection patterns. And all these measurements were conducted in vitro. So our finding of hub neurons is, to the best of our knowledge, the first demonstration of the existence of hub neurons in the living brain. So finally, what about the possible role of hub neurons in the motor cortex? So we measured the relationship between the number of connections and the strengths of directional tuning. We found that hub neurons tended to have less prominent directional tuning compared to less connected neurons. So I don't have the time to show you, but we also found that while hub neurons were less sensitive to movement direction, they were actually more sensitive to the timing of the movement. And there were also had a higher correlation between the activity of hub neurons and the activity of neighbor neurons. So this suggests that a possible functional role of the network hubs in the motor cortex is to synchronize local dynamics and potentially serve as local conductors of the neural orchestra. So to sum up, we developed a rapid optical method for mapping effective connectivity in vivo. We combined this method with new behavioral paradigms that reveal directional tuning in the motor cortex of naive untrained mice. And this directional tuning in the motor cortex had a global salt and pepper organization with local clustering of neurons with similar directional properties. We found potentially mini columns in the mouse cortex with like-to-like connectivity and a Mexican hat-like profile of excitation and inhibition. And we also demonstrated the existence of hub neurons, which might potentially act as pacemakers of the motor cortex. So if you want to hear more, you are welcome to come to the workshop on Tuesday. I'll talk more about that stuff and about maps for action selection in the frontal cortex and attractor mechanisms for their generation. And finally, a little advertisement. I opened a lab just recently in Tel Aviv University, where we're going to combine optical interrogation methods for circuit analysis and mesoscale imaging. So if you are interested in studying the link between network dynamics and behavior by reading and writing information to the brain, please reach out to me, and I'm happy to take questions. Thank you.
Nothing on the app. All right, take the first question.
Hello. Hi, nice talk. So I'm just interested in a couple of technical things. You show a very nice video that you just excite the neurons. What are the limitations? How many neurons you're exciting? You just go one by one, or 10, 20, 30? And what is the effect in your hub neurons? And especially, what is the diversity of the animals? If you go with number 1, 2, 3, 4, what is the diversity of the hub neurons? What can you tell about that? Thank you.
Yeah, thanks. So we are currently stimulating about 400 to 500 targets in the upper plane with image from several volumes. And basically, we look at combinatorically at responses at the rest of the population, which comprise about 2,000 neurons. So we are essentially getting a kind of pretty sparse functional connectome, if you want, from this population of about 2,000 neurons. So the edges, the output degrees which we define, which we use to define the hub neurons, are basically to some extent limited by this sampling that we have.
Take one from the other side.
Thank you. That was fantastic. So you do a lot of optogenetic stimulations, right? A lot of different places. And I imagine that this might have some effect on the brain. You might be sending a lot of energy there. You might be heating up the brain. Is there any evidence that you saw that there was an effect of the stimulation? Like, did you see any different non-natural behavior, or did you see any damage?
Yeah, so we had to tune the stimulation parameters, indeed not to overstimulate the brain. If you do it with stronger stimulation patterns, you indeed can cause neuronal death. So in the regimes that we are using, we are actually able to return to the same population of neurons across days and stimulate them repeatedly. So at least at this level, we don't see any damage.
Thank you.
Yeah.
Time for one more.
Hi, great talk. I was wondering whether the hub neurons are excitatory or inhibitory, and whether you can speculate a little bit more about their functional role.
Yeah, so far we only were able to assess excitatory connectivity here. So we are stimulating excitatory neurons, and we read out the response of other excitatory neurons. So whatever inhibitions that we see is actually has to be polysynaptic, and that's one of the reasons why we think it's tiny. So there have been in vitro studies about inhibitory hub neurons, but we don't assess them right now with this variation of this method.
Thank you.
All right, in the interest of time, I think we'll move on to our next speaker. If we can catch up with Arseny later at the workshops, we'll have extra time. All right, so our next speaker is Laura Bella Nouwen, and she'll be talking to us today about invariant neural subspaces maintained by feedback modulation.
Yeah, hello everyone. I'm going to wait for this to show up. In the meantime, yeah, my name is Laura Bella Nouwen. I'm a PhD student in Henning Spreekler's lab at the TU Berlin. And I'm really excited to be able to present our work, some of our recent ideas on how invariant neural subspaces can be maintained by feedback modulation. And I hope this will eventually show up. Yeah. I'm going to try to disconnect and connect again. 1 second. I don't know.
Try does the other one work?
I mean, it worked before, so I'm not sure why it doesn't work now. Okay, I'll try to switch adapters. Sorry for this. You all get a chance to breathe and think about your own stuff. Let's see. Okay, there we go. Okay, sorry for the delay. Yeah. I just want to point out that this is joint work with my awesome colleague Joram Kaiser, who's also at this conference, and under the supervision of Henning Spreekler, who's also here. Right. So the classical idea of sensory processing is that sensory stimuli are processed hierarchically, right? And that's because there's a consistent feed-forward motif that we can find across sensory systems. And in this classical idea, in this classical view, the idea is kind of that you can extract increasingly complex features and thereby get a representation that is increasingly useful for behavior. And these sorts of models have been super successful, specifically with the rise and the re-rise of deep networks. And one of the success stories here is that of invariant processing, and in particular, invariant object recognition. So the idea here is that at kind of lower levels of this hierarchy, representations are really fragile and highly entangled, such as in V1. But then at higher levels of the hierarchy, you get representations that are sort of invariant across variations of a stimulus and allow a linear separation of distinct objects. But of course, the brain is not a purely feed-forward system. There's an abundance of feedback connections, even at sort of areas considered lower-level sensory. And interestingly, the effect of this feedback is mostly to modulate rather to drive sensory responses. So we were kind of wondering what the function of these modulatory feedback connections could be. And in particular, in this project, we explored the hypothesis that feedback modulation could serve as a neuromechanism for invariant perception. So we're a computational lab, which means that our approach here is to use tricks from machine learning to optimize feedback in biologically constrained networks. And then as a next step, we kind of reverse engineer how these resulting networks work. So as an example of invariant processing, we use a task that is inspired by the cocktail party problem. So imagine you're in a room where a lot of people are talking at the same time, and you're trying to listen to one specific person. So I think we can all relate to this problem at the moment. We can also call it the post-accession problem. So the basic idea is that you have a couple of unknown sources that are mixed together to generate the sensory stimuli. And now what you want to do is to somehow process these stimuli to kind of recover these sources. Now, we give this task a little twist. You know, people at a post-accession can move around, right? Which means that the mixture of the sources can actually change over time. And this gives us a context that is actually dynamic in time. This is what we call the dynamic blind source separation task. So this can't really be solved with a feed-forward network because different contexts require different feed-forward processing, right? So we somehow need to infer what the current context is from the input data and then kind of adapt processing to maintain an invariant output. Now, what I will show today is that we can achieve this with a modulator that receives information from the feed-forward network and provides a modulatory signal to the feed-forward processing. Right? So we model this modulator as an RNN, and we train it on this task. But for the remaining time of the talk, I will not focus on the modulator itself, but on the effect that it has on the feed-forward network. So as a proof of concept, we use the simplest possible feed-forward network we can think of, which is just a linear readout of the input stimuli. And this readout here is multiplicatively modulated by feedback from this modulator. And ideally, we would want that this kind of product here is equal to the inverse of the mixing, right? Because we want to undo this mixing, okay? So let me show you this model in action. As sources, we use signals that look like this, so it's kind of like combinations of signs. And the sensory stimuli then look something like this, so it's just a mixture of these sources. Now, after we've trained this network, the output nicely matches the sources. Okay. So now what happens when we change the context? Well, the sources are still the same, but the sensory stimuli sort of change. And now after a very brief error that you can almost not see here, but is there, our network output again matches these sources, right? This error occurs because the modulator first has to kind of infer what the current context is and then adapt the modulation it provides in response to that, right? So actually in this case, what the modulator does is that it kind of adapts these readout weights such that they invert the current mixing. And you can also see that the modulation here changes on the time scale of the context. So to quantify this, we look at the correlation of the sources with the stimuli and with the output of the network. And ideally, we would want that each output correlates with only one of the sources, right? So we take the mean absolute difference of the two, and this is what we call the signal clarity. So this is what I'm going to use to measure performance in this network. So the signal clarity for the sensory stimuli, that's around 0.5, and we can use that as a control. And for this example, the signal clarity measure here is close to 1. This is what we would want. So, but now, what happens if we introduce some biological constraints to this model, right? So what about the time scale of modulation? The modulatory signals are typically kind of thought to be more slower signals, right? So we look at networks that had increasingly slow feedback signals, and what we found is that they're actually not very sensitive to the time scale of this modulation. So in principle, this modulation can be slow. Now, but what about the spatial scale, right? In this particular example, the modulation modulates distinct weights, synapses, axons, whatever. Like, that's not typically how we think about modulatory signals. So to address kind of the spatial scale, what we do is that we project these sensory stimuli into a population of neurons. And then from this population of neurons, we take a linear readout to get the output. Now, what this allows us to do is to model sort of diffusive modulation in which neighboring neurons are modulated similarly. And now, after training this network again, we can look at the modulation to all the neurons over time, and we find that they're actually modulated in groups. And again, that this modulation switches on the timescale of the context, and this is what allows us to solve the task. So now, by changing the number of feedback signals, and also this kind of the width of this modulation, we can control the spatial extent of the modulation. And what we found is that for this particular low-dimensional task, we only need a low number of feedback signals, and this modulation width can actually be pretty much arbitrarily broad as long as it's not uniform across neurons. So for this particular mechanism that we suggest here, the sort of biological constraints that we would observe in the brain of neuromodulatory mechanisms are not really a problem. But now, to the interesting stuff, how does this invariance even emerge, right? So does it come from the single neuron level, or does it only arise at the population level? So one option would be that, depending on the current context, distinct kind of populations, subpopulations of neurons represent distinct sources. And what the feedback does is that it controls which of these neurons can kind of contribute to the output. Alternatively, we could imagine that all neurons have sort of a mixed representation of the sources, and the feedback kind of regulates how they work together to create the output. Now, to test this, we first look at the signal clarity of sort of single neurons in this feed-forward network in different stages. As we would expect, the signal clarity for the sensory stimuli is low and is high for the network output. But we found that for this neural population, it's also actually low. So there are some neurons that have a higher signal clarity in some context, but there's not really a consistent group of neurons that has a high signal clarity, which means that at the single neuron level, these sources are really not separated, and there's no invariance at the single neuron level. So the invariance must somehow arise at the population level. And to kind of verify this, we look at whether we can decode the sources from different stages of this feed-forward network. And we found that the decoding accuracy was as high on this neural population as in the output, which is maybe not surprising because what the output does is take a linear readout of these sources, right? But this shows that we can find this context invariant subspace. But how is this context invariant subspace maintained by the feedback modulation? And specifically, we were wondering if we can disentangle kind of the effect of the context changes and the role of feedback, because that would usually occur at the same time. And now the cool thing is that this is a model, so we can actually do this by freezing the feedback in the initial period after a context change. So we simulated an experiment in which that consists of 3 stages. So first, for a given context, we run the model and feedback is intact. Then we change the context and we freeze the feedback. So this impairs the network's performance. Now then, we have the same context and we let the feedback do its job, right? So it again performs well in this task. And now we looked at the population activity in a 3-dimensional space made up of the first principal component and the 2 readout axes. And we find this pattern here lives in a 2-dimensional subspace. And if you project this down to only the readout axis, you see that this pattern here matches the sources. So when we now we change the context but freeze the feedback, what happens is that this pattern stays in this 2-dimensional subspace, and it's sort of like rotated and stretched, right? Which means that it does the projection to the readout doesn't match the sources. So now we were wondering if the feedback would just like undo this transformation, would it just reverse this? But actually, that's not the case. We found that what the feedback does is that it completely reorients this whole subspace, and this is what enabled the output to kind of still match the sources and realign this whole thing with the invariant subspace. We also quantify this by looking kind of at the angle of the original subspace with a subspace for context changes and for feedback changes. And we found that consistently, this feedback created a reorientation. And interestingly, this reorientation here, the magnitude of it depends on whether the new context is sort of close or far from the initial context in our kind of continuous context space. So now finally, just as a small sneak peek, we also found that kind of these findings generalize to more complex networks that have that obey Dale's law. And we have some ideas on how this feedback modulation relates to layer 1 interneurons. And also we found that the context cannot necessarily be decoded linearly, which we think is really interesting for how we look at neural data. So if you want to know more, you can find me offline. I'm also at the workshops, or you can look at the preprint online. We updated it a few days ago. So now I want to summarize. What we found is that this feedback modulation can indeed serve this function of context-invariant sensory processing. And the feedback in our model can be slow and can be spatially diffuse. The invariance only arises at the population level, not the single neuron level. And the role of feedback here really is to dynamically reorient the population representation, which allows us to maintain an invariant subspace. So according to our model, manipulating these feedback projections should impair context-invariant subspaces. And we think that here population-level recordings and analyses may really be necessary to uncover these mechanisms of invariant processing. With this, I want to give a shout out to my lab. I'm really very grateful that I was able to present this work on the stage, and I want to thank you for your attention and open for questions.
Any questions? Kevin?
That was great. I have a question about training and testing. Did you test your network on contexts that were novel?
Yeah.
And if they were novel, were they interpolated or extrapolated from the
Yeah, okay, that's a really good question. So I didn't really have time to go into that. Usually we tested and trained on the same kind of distribution of context, right? So it's a continuous context space that is two-dimensional because we normalize these mixing matrices in a way, but that's not so important. So we kind of sample new random contexts every time, which means that it's unlikely that you get exactly the same context again, but the network kind of has to interpolate between the data points that it has already seen. And we actually found that since this modulator there has to learn some sort of internal model of the task, right, of like what the context could be and also of the sources, is that if you use contexts that are completely different from anything that we've used during the training, this can be an issue for the model, but I think this is something that we would expect. Yeah.
All right, I think in the interest of time, we'll have to move on to the next speaker. Right, our last speaker in this session is Mitra Javadzadeh. Okay, thank you. Who's going to talk to us about the dynamic causal communication channels between neocortical areas.
Hello, everyone. My name is Mitra. I'm a postdoc at UCL, and today I will be showing you some of our work on interareal communication in the neocortex. So we are interested in understanding how different brain areas influence each other and how the neural responses that we would record in a part of the brain is being constantly shaped by the activity that is going on in all other parts of the brain. So to understand these interactions, we studied the visual cortex of mice as a model system. And specifically today, I'm going to talk about the feedback influence from higher visual area LM on area V1. You can think of LM as the mouse equivalent of V2. So let's say we take a mouse, we show it a visual stimulus, a grating, and we record in these areas. And we will see activity in response to the visual stimulus. And our question is, what is the contribution of LM in shaping these visual responses that we see in V1? So we have an approach to answer this question. We use optogenetics to briefly silence the activity in LM, as shown here in blue, and then we ask what is happening in V1 as a result. So this way we can map the causal influence of LM on V1. And also we want to understand communication on a population level. So we record with single cell resolution from both areas using silicon probes, so we can see how the activity of each individual V1 neuron is being changed when we silence LM. So we are using this pulse of light as a way to probe communication at a particular time point during visual processing. But we also want to see how these interactions evolve over time and shape visual responses in V1 throughout the whole visual processing. So what we do is that we keep showing the same stimulus to the mouse in multiple trials, and from trial to trial, we change the time of silencing LM. So this way we can map communication in all these time windows over the time course of visual processing. And to have a sense of the time scales, the whole visual stimulus is on for half a second. We are tiling this duration by 8 silencing time windows, and we have a 70 millisecond temporal resolution between the onset of these temporal silencing time windows. So now I'm going to show you some results. This is an example neuron in V1. In black, you see the activity of the cell in control trials, and in blue, when we silence the activity in LM in this time window T1. And you see that when we silence LM, the activity of this V1 neuron is not influenced. But if we silence LM a little bit later at T2, now we see an influence on the firing of this cell. And if we silence even later, again, there is no effect. So the influence of LM on this V1 neuron was confined to this very brief time window, and nothing is happening even 70 milliseconds before or after. And notice that these responses are reliable over trials. It's just that they're very brief in time. So this was a surprising finding for us. And it turns out that if we look at all V1 neurons, this is a typical behavior. So across all V1 neurons, if the neurons are influenced by silencing LM at all, they're influenced in only 1 or 2 time windows. So the time, basically the windows of influence are very brief. But the exact time of this influence could be any of the 8 silencing time windows that we looked at. And interestingly, if we pool together all the silencing effects that we see in V1 over all V1 neurons and all the time points, we see a lot of diversity. We see some cells are suppressed, some cells are excited, but they average to a very small number, around 0. So this emphasizes the importance of having this axis of time and single cells to understand the structure of interareal influences. Otherwise, if you just average everything, we will see a net 0 effect. So to get a holistic view of communication over all neurons and time points, we are going to look at the population activity in V1. So let's consider the activity at a particular time window, and when each point is showing one trial. And as the visual responses evolve over time, these points move in the activity space of V1. Now, at each one of these time windows, when we silence LM, we are moving the activity to the corresponding blue points. And now we can ask first, what is the magnitude of this displacement at each time point? And what is the direction? And these directions we call communication directions at each time window. And they specify, in a way, a pattern of activity, no, a pattern of influence from LM on V1 neurons at that particular time point. So if we look at the magnitudes of influence on V1, we see that they don't change significantly over time. However, the directions of influence are changing all the time. So here I'm showing you the dot product between a pair of communication directions that are calculated at 2 different time points as a function of the time lag between them. So you see, as time goes on, these communication directions get more and more dissimilar, meaning that the patterns of influence on V1 are changing over time. And this is very fast. The time constants of this decay are around 100 milliseconds. And for comparison, I can show you what happens to the activity itself. So if you look at the directions that specify the patterns of activity in V1 or LM in control trials at each time window, we can ask how fast do these directions change over time. And the answer is that on the time scales that you're looking at, they're not changing much. They're relatively stable. And we have done tons of controls and simulations to show that these differences between communication and activity is not explained by the differences in the magnitude of these vectors or different levels of noise. So our first population level insight here was that communication directions are changing all the time, while the activity patterns are stable. We also wanted to see how the behavioral context changes communication between areas. So we trained mice to do a visual discrimination task. So they see 2 types of gratings. One of them is rewarded, the go stimulus, and the other one, no go stimulus, is not rewarded. The mice are not required to do anything, and they don't even receive a punishment. What I've shown you so far was what was happening when the mice were processing the no-go stimulus. And here in green now, I am overlaying what happens during the processing of the go stimulus. And you can see that the communication directions are now changing at much faster rates. So they become completely dissimilar even one time point later, while the patterns of activity are changing at the same rate. So not only the patterns of communication are changing all the time, but the speed of these changes is a function of behavioral context. They can accelerate or slow down dynamically depending on the context. So after having these experimental observations, we asked what is the mechanism behind them. So at the moment, we are doing some modeling work to understand the mechanism behind these fast changes in communication. So when we silence LM, what we are doing is to push its activity to 0. And we saw that the patterns of LM activity are stable over time. So when we are silencing LM at different time points, we are causing a push in the same direction in LM. However, what we see in V1 is that V1 is influenced in completely different directions at these different time points. So how does this happen? One possibility is that you need a nonlinear transfer function from LM to V1 to explain what is happening in V1, arising from things like synaptic mechanisms. But a second possibility is that you might be able to explain what is happening in V1 by a linear mapping from LM that selectively amplifies specific features of LM activity. So we see these vectors are almost the same, but they're not exactly the same. There are some differences. There could be some high-dimensional structure here that explains very little of the variance, and we would normally discard it as noise, but these features could be exactly spanning the subspace in LM that is relevant for influencing V1, and these features would therefore get amplified when mapped to V1. So we are asking, could a linear model capture the time-varying influences that we see in V1? And if this is the case, perhaps these models could give us some generic insights about what features of activity in a cortical area are relevant for influencing other areas. So we are using linear dynamical systems with Poisson observations to model the activity in both V1 and LM over time. So we have two sets of latent variables, V1 states and LM states, such that the observations, so basically the neural recordings that we have in V1 only depend on V1 states, and the observations in LM only depend on LM states. And these states are evolving over time with linear dynamics. And this dynamics matrix A here, we are not constraining this matrix when we are fitting the models. So the idea is that we are capturing all possible temporal dependencies between all V1 and LM states over time. So basically, we can capture both local and long-range influences between the states. And we also have external inputs. We have the visual stimulus that is influencing both V1 and LM states, and the optogenetic silencing, which is only influencing LM, and some Gaussians. So we are still working with different varieties of these models. We don't have anything conclusive, but there seems to be early indications that these models could capture some of the features of communication. So here I'm showing you one cell that is only transiently affected. So this is a cell in V1 that is only transiently affected when we silence LM at time point T2 and not T1 or T3. And this is the prediction of the model from actually the activity of LM, what happens in V1. And you see that the model can capture this transient effect. And finally, besides looking at the mechanisms, we are also thinking in the direction of the potential functions of this regime of communication. So LM is influencing V1 along dimensions that are constantly changing over time. What could this be good for? So you can think of different directions in the activity space of V1 as directions that are encoding different variables or representing different linear readouts from V1. And the fact that LM's influence on V1 is changing all the time means that these influences could transiently align with various coding or readout dimensions in V1, and it would mean that LM could selectively influence a particular readout from V1 at any time point. And this would mean that the function of LM to V1 feedback could be flexible in spite of the activity itself being slow in both V1 and LM. As an example of that, we have looked at stimulus discrimination in V1. So we are asking, looking at the population activity in V1, how well can we decode the identity of the stimulus that the mouse is seeing? And it turns out that silencing LM changes this stimulus discrimination in V1 only during a brief time window in the beginning of the visual stimulus. And this is the same time window that we see a behavioral effect with silencing. And this is interesting because if you remember, the magnitudes of LM's influence on V1 were not changing much over time. So this transient influence would mean that during this time window, the patterns of LM influence are aligning or overlapping with the patterns that are relevant for stimulus decoding, stimulus discrimination in V1. And then after this perhaps behavioral relevant time window passes, these directions rotate out of alignment, and LM won't be able to affect stimulus coding in V1 anymore. And with this, I will end. Thanks for listening.
Very nice talk. First question about the precision of your experiments. So I may miss that part. In the silencing experiments, are you locally injecting the probe, or you have used a transgenic animal?
Yeah, good question. So we are locally injecting the opsin, so we are limited by the size of the injection. Yeah.
Okay, and then for the stimulus for the first part, as we know, V1 and LM has different spatial and temporal frequency preferences.
Yes.
So do you see the effect of the stimulus on the optogenetic silencing, where the effect is actually, a lot, or not actually, changing at all? Because you show that depending on the timing of the silencing, you have a differences in control versus the optogenetic silencing. Does it really depend on the stimulus type, or the spatial and temporal frequency of the stimulus?
So I can answer it in two parts. First of all, the preferences are of V1 and LM, although there are some differences, but they're very much overlapping.
Sure.
But on a similar line, we have actually looked at if any properties of the cells in V1, such as tuning to the stimulus, can explain how much they're influenced by LM. And interestingly, we don't find any pattern. So it seems that there's no proper, it's more of a homogeneous effect that affects all the cells, and we can't find any pattern in the cell type or preference or which cortical depth they are at.
And for the LM injection, last question, is it confined to layer 2-3, or?
No, all the depth of the
all
cortex.
The depth. Okay, thank you.
All right, I'm sorry. I think just because we're running way over time now, so we'll have to cut off. Sorry. Thank all of our speakers from this session. And just a reminder, there is a diversity luncheon. So those of you who have want to go to that, it's over at noon in the hotel, the Valegia Opera Hotel, which is just a few blocks over. All right.
tool.